Good afternoon and welcome back, everyone. My name is Lisa Vanderlaan, and I'm a first-year plant breeding PhD student in Dr. Danny Singh's lab. I'm happy to introduce this afternoon Dr. Guiping as our first speaker for the afternoon session. Dr. Hu is an associate professor in the Department of Industrial and Manufacturing Systems Engineering here at Iowa State University. She's also affiliated with graduate programs in Sustainable Agriculture, Bioeconomy Institute, and Bioinformatics and Computational Biology. Dr. Hu's research has focused on operations research and data analytics with applications in renewable energy systems, supply chain design, bioinformatics, and sustainable agriculture. Her research has been supported by NSF, USDA, DOE, and DoD, and she's published over 100 journal papers or conference proceedings. Dr. Hu is also active in serving the profession of industrial engineering, including as an associate editor and editorial committee, and as an elected officer for IISE and Informs. This afternoon, Dr. Hu will be presenting about systems design thinking and data analytics in plant breeding. Please join me in welcoming Dr. Hu.
Thank you, Lisa, for the wonderful introduction. Let me share my slides. Can you hear me okay? Okay, perfect. Get to the presentation mode. I assume you can see the slides, right? Okay, all right. If there is any technical issue, just let me know. Great. Okay, so again, thank you for the invitation. This is a great event. I was able to attend a couple presentations in the morning, Dr. Mark Cooper's and part of Dr. Javier's talk. So those were great. I had some meetings, so I was not able to stay. But today, I like to talk about some work that I have been doing in the last 4 or 5 years that we've used machine learning and system design thinking as it applied to plant breeding, but also probably a little bit broader than plant breeding. Because I think in order to make, in order to make sense of plant breeding, I think it's important to consider the context of agricultural production in general. See my slides. It cannot. Oops. Let me see. Okay, so I want to send some acknowledgement. As Lisa mentioned earlier, this work is primarily supported by NSF and USDA, and I'm also one of the Plant Science Institute scholars. So PSI has been providing some funding as well as support. We have been meeting on a weekly basis to discuss research. That had been providing tremendous support for me as an engineer to conduct work in the plant breeding as well as agricultural production area. And also some of the work is in collaboration with Syngenta. Yeah, let me get the pointer out. And these are a few students of a PhD student of mine who have been contribute to the work that I'll be talking about. And in terms of the faculty collaborators, I have been working with Pat Schnabel in the agronomy, and Li Zhiwang, who is also an engineer and a PSI scholar as well. Soterius, who is a faculty in the agronomy, is actually collaborating on the very next piece of work, which I'll be talking about, integrating or connecting crop model with machine learning models. And then Bill Beavers has been very helpful in terms of helping me understanding a lot of plant breeding concept. And he has been very supportive in terms of bringing system engineering as well as operations research to plant breeding. So before I talk about the specific project, I want to share a couple of sort of big picture slides because when I was talking to Lisa and some other organizer of this event, they asked me to talk about system design thinking as well as data analytics, right? So my background is in industrial engineering. So I was trained as a systems engineering. What does that mean? That means when we view things, before we zoom, we tend to zoom out. And in terms of data analytics and optimization, which is somewhere equivalent to operations research. So basically, their decision-making component there. So when I view the plum breeding, we are trying to achieve some superior phenotype, whether it's yield or some type of trait that we are really focusing on. It's actually determined by the genotype and environment. Sort of the G by E, which was discussed earlier, right, in the discussions. And what I want to throw in over here is the management piece. The reason is that I think management is really the component that can be involved in decision-making and system decision design, okay? And in terms of the data analytics, I view there are 3 levels. The first one is called descriptive. What does that mean? That basically means we're trying to understand what happened. We have historic data. We're trying to fit some model to explain what was happening in the past. And then the next level is called what's called predictive. So given what happened in the past, our next task is what is going to happen in the future. For example, yield prediction. Or if we were to plant this seed in this new environment, what is going to happen? Can we expect a drought resistance? And then the very next level, which I believe is an important level, is what's called prescriptive, right? So what prescriptive level analysis doing is really provide the decision support. Because the first 2 levels, we're trying to understand what happened and what is going to happen, right? But in order to make a difference in either farmers' decision-making or other agricultural decision-making, in order to combine the data analytics to impactful decision, we need to go through this prescriptive analysis. Okay, so feel free to interrupt me if there are questions, okay? And then another slide which I want to talk about is precision agriculture, because our topic today is on plum breeding. But I think precision agriculture is really involved and should be integrated in the breeding process. So just one quick slide. I know that we are going to dive into the specifics. But it started from the concept of farming by soil. And some of my work has to do with precision comprehensive management. We have worked on papers on irrigation planning. I know in Iowa we don't need to irrigate, but for example, in Nebraska, in California, there is need for irrigation. And seed selection, when to plant, what to plant, fertilizer application. Do we do pre-season, during the season, and then when should we apply it? How much do we put in? Is it manure or is it nitrogen fertilizer? In what kind of format? Pesticide application. And another one is what's called vehicle routing. When I was talking to some of the farmers, they said, well, machinery management is another important task they need to deal with during the growing season. And the very last one I put in here is what's called environmental impact analysis. So I come from a background of, decision making, and in the decision making theory, there is this concept of trade-off analysis, multi-objective decision making and optimization. So it's very important to think about when we are making decision in agriculture, in plum breeding, what kind of trade-off, right? And environmental impact analysis and trade-off between profitability as well as sustainability is something we really need to think about. I know Dr. Javier talked about the work that he was doing with Corteva. I know Corteva has some internal tools, but I believe what we are doing here can maybe enhance and maybe provide some additional quantitative support and maybe some additional maybe risk and profitability trade-off analysis over there. And what I planned for today is that I'm going to talk about sort of 3 topics. And I won't talk into too much specific technical detail, but if you're interested in any of those, feel free to contact me. I can share a paper, we can talk about this offline, but the purpose of this talk is really introduce some concept, okay, and really generate some discussion, okay. I'm by no means an expert in plum breeding, okay. I'm still an elementary school student trying to learn how the techniques we develop in engineering, in decision science, in data analytics can help this community move to the next level. So the first one that I want to talk about is a work that collaborated with Sotiris. In this work, we are trying to couple machine learning and crop monitoring and to achieve better yield prediction. And we all know that there have been advances in machine learning and crop monitoring, which was talking about earlier in talks. And each of them will offer unique capabilities. And oftentimes, so far, they have been studied or used separately. And the hypothesis we are trying to test here is that if we combine them together, by having a hybrid model, this is where the system analysis can come in. If we combine them and try to bring in the merits of these two approaches, can we achieve better performance? So this is one slide to talk about crop monitoring as well as machine learning. There are pros and cons of each of them. For simulation modeling, crop monitoring, APSIM as an example, they have achieved reasonable prediction accuracy, but some of the downside include it will require user skill to calibrate the model, try to understand the different inputs, and then the computational time tend to be long and their data storage constraints. In terms of machine learning, some of the pros include, well, there are a lot of improvement in the community of computer science as well as data science. And we can deal with linear, nonlinear data and also non-normal data. The prediction performance tend to be high if you have enough data. Okay, now some of the problems include if you don't have enough data, then the prediction accuracy may not be very high. And another big problem is that there are some explainability problem or model transparency problem, especially when we are talking about neural network-based model, right? So those tend to be a black box, but we've developed some tools that can kind of dive into the box and try to improve the transparency. But that's not what I'm trying to talk about today. So in the work of integrating crop modeling and machine learning, try to improve yield prediction, the type of data we are using is same, yield data, weather data, soil and management information data. And if we're taking the machine learning route, you basically just put the data in the machine learning model, let the model run, crank, and then you'll get the yield prediction out. For crop modeling, you'll use this type of data to calibrate the model and you'll generate different types of output of interest. For this specific study, what we are doing is that we are using crop modeling to generate some intermediary traits. And those traits will be used as an input or features for the machine learning. Okay, so there are two reasons for that. First, when you generate this, you are including additional information in the growth stages. And second, you are trying to improve the transparency of the model by actually incorporate additional information that are not typically included in the machine learning model. Okay, so some of the research questions we are trying to address. First, does this hybrid approach result in better yield prediction? In this specific study, we were using corn, but we also did for soybean. And this can be used for other type of crops as well. And which combination of the hybrid model, meaning that what kind of features we should use, what kind of model should we use? When I talk about machine learning models, there are all kinds of models out there. And you can have integration of model like what's called ensemble model on top of it. And then the next question is that what features, what kind of input are critical for this type of analysis? Okay, so we used APSIM. And some of the APSIM features include phenology-related, flowering time, maturity date, and crop-related. I'm not going into too much detail given the time. And then soil and weather-related. Okay, so these are APSIM features that are generated from the crop model. And then in terms of the data set, we used USDA data set, and then the features are included over here, which include input from APSIM, the plant population, planting date, weather information was from DayMet, and then soil information as well. The target is of course corn yield. We did it for 3 states, but we are doing it for the whole Midwest, 12 states. So there was some data pre-processing in this analysis. I'm not going into too much detail about this. So there are feature construction, feature selection, as well as model selection. If you're interested in knowing more about this, I'll be happy to provide detail offline. So data processing is actually very important, which was mentioned earlier in the talk by Mark as well as Javier in terms of, garbage in, garbage out, right? So data processing, in this case, we first did just plot the trend and try to understand county level, what are the variations. And then we did some feature construction in terms of yield trend as well as historic yield average. And by doing that, we were actually trying to capture some of the information that tend to be overlooked if we just blend it, throw everything into the machine learning model. So we constructed some feature. And I want to quickly show you some results. Remember the 3 research questions we're trying to address. We have the benchmark, which is on the top, the blue line, and then the orange line here is the hybrid model. And then the axis over here, we analyzed a variety of machine learning or statistical learning models. The first 5 over here, lasso, XGBoost, light GBM, random forest, and linear regression, those are sort of more standalone models. And then the rest of those are actually more of using optimization or ensemble on top of it to try to combine the different models together. Okay, so the key point I want to show, and then the y-axis is the relative root mean square error in terms of percentages. The key point I want to talk about is that when we incorporate those APSIM, the prediction error or the test RMSE, okay, relative root mean square error dropped significantly across all the models, regardless of the model you are using, okay? And this is the average of the testing year 2017 and 2018. So that's actually quite substantial. And this some additional plot. Here we have the top row are the benchmarks in terms of the standalone machine learning model. And then the bottom is the hybrid, okay? And then the each of the column is representing one type of model that we used. As you can tell, when we are going from the top to bottom, the improvement is quite obvious in terms of the points are more aligned with the y equals x-axis, the 45-degree line, which is showing we are predicting very close to the x-axis is the actual, y-axis is the predicted. So the 45-degree line is what we are trying to get close to. Okay, so this is just additional demonstration. And we further did, what if we just include phenology related, crop related, or soil weather related? The previous analysis was, what if we have all 3 of those? Now, if we just consider one categories, okay? And we did some analysis on this. And another analysis we did is variable importance for the APSIM additional feature. Okay, so here the different color bar representing the different years. So the salmon is 2017, and then the gray is 2018. The key message I want to deliver over here is that average drought resistance and also average water table in the season, those are the top 2 important features. And as you can see, across the year, they tend to be relatively consistent. And this is not based on just one simulation run or one analysis. We've done rigorous permutation analysis to look at a variety of scenarios. And to conclude this piece of work, the key point is that when we add additional APSIM input, so when we integrate machine learning and crop model, the prediction accuracy have been increased substantially, up to 29%. And then the most important APSIM features include soil and then basically drought related features. And that's just the same information, additional soil weather variable can further improve this because we only have a limited number of them. Are there any questions about the first piece of work? Because I have a total of 3. Or maybe we can save it for later if that works. So the next one is a work that a PhD student of mine is doing with Darren Muir from plant pathology and Yuba in the same department. So in this study, we were using what's called a gated recurrent unit, which is similar to recurrent neural network for soybean sudden death syndrome detection with satellite imagery. So you may be wondering, well, why are you talking about image analysis, right? Again, I'm trying to get to some system view because when I was talking to the soybean farmers, this is one of the most important things they care about or they worry about, plant disease. And without going into too much detail, paying attention to the time, is that it's a challenging problem when you are trying to detect when the disease, how severe that disease is, because it's variable in time, space, and also genotype. Now, some of the sensing technology include thermography, fluorescence. I'll just go through this relatively quickly because these are background information. And the research problem, what we are trying to address here is that traditionally, this detection has been what's called near sensing, which is you take a closer look at the leaves and detect whether there is disease. And what we are trying to do in this study was talking about remote sensing, because from a system analysis perspective, in order to really make this useful, you really need to zoom out and think about how can we apply this when we have a big field, right? When we have multiple fields. And in terms of remote sensing, there are UAV and tractor-mounted tools as well as satellite. In this specific study, some of the results I'm going to show you, these are based on satellite. This was the first piece of work that we did. The paper has been published, and then we are right now working on some UAV data as well. Satellite imaging, there are some benefits, low marginal cost and continuous monitoring. Of course, there are problems for them as well because it's far, the resolution tends to be low, and also there may be some, for example, clouds or some other factors that may come into play. So here I put higher resolution because if you pay, you can actually get a higher resolution, but you need to work with specific company ahead of time. So in terms of this work, the workflow is first we collect the data through satellite imaging, and then we'll connect that to disease information, and there is data pre-processing as well. And sometimes there is data visualization to further understand how we'll pre-process the data and try to do feature selection and feature engineering. And then this is the analysis. Now, in terms of the data, we were using this data provided by Darren, his research group, and this is information from 2016. We also did it for 17 and 18. And the information we got was the RGB information, and then you can calculate the NDVI, normalized difference vegetation index. And we also consider rotation information. What was planted there ahead of time. In some follow-up work, we are trying to incorporate some additional information, some management information, because that can help us understand the disease. Predictor as well as output, this is a classification problem. So when SDS rating is about 5%, we'll classify as 1, which means it's diseased, otherwise it is not diseased. And these are the dates that we were looking into. And in terms of the research method, previously people typically look at statically, individual, right? So when you have this picture there, we're trying to detect whether there is disease or not. But if we have time series, time-stamped data, then in addition to detecting whether this piece of picture has disease or not, you can also look at time series, right? Because for SDS, if there is disease, then it will be diseased for the rest of the season. So we have designed sort of two categories of models, sequence-based as well as non-sequence-based, and we're trying to compare these. The sequence-based is a recurrent neural network based, which is called gated recurrent units. And then the non-sequenced, we have XGBoost and fully connected neural network. So, so this is just a picture of the gated recurrent units. I'm not going to go into much detail about this. And for fully connected neural network, and XGBoost, those are more traditional machine learning models. And one quick slide to show the improvement or the differences between the proposed model, which is the GRU gated recurrent unit, and then the XGBoost, and then fully connected neural network. Something I want to mention here is that we also analyze other models, but we picked these two machine learning models because those are the two that perform the best. So as you can tell, see from this table, we did a variety of experiment analysis. And the gated recurrent unit, GRU, were able to improve in the machine learning or in the data analytic area. This is actually quite substantial because the accuracy was already quite high to begin with. Okay, so to summarize on this little piece of work, we designed a gated recurrent unit for soybean SDS detection. And this has improved upon the traditional models of neural network. And we also did some prediction work, which I did not include in this presentation, given the interest of the time. Okay, so the last sort of category of models I want to talk about is, this is a collaboration with Pat Schnabel and Li Zhiwang. And in this work, what we were doing, this is probably closer related to plant breeding. So what we did was simulation optimization and reinforcement learning techniques in genomic selection. And I will elaborate that in the next few slides. So I put one slide over here. I know I'm probably preaching to the choir because this group, you're just focusing on genomic selection, not genomics, plant breeding in general. So you probably know this better than I do, okay? So typically phenotypical selection, you just look at the trait and you decide which individual should be advanced. And genomic selection, what genomic selection is doing, that you first genotype and you look at the genotypical information. I'm using the 0-1 to demonstrate the genotypical information. And then you do some predictive type of analysis, like the talk, some of the talk mentioned earlier in the morning. And then based on your prediction, you select the individual to be advanced to the next generation. Okay, the typical genomic selection is what's called truncation selection. So supposedly, given the 0-1 information, and I predict the plant, this is this high, and this one is this tall. Okay, so they typically do what's called truncation selection. So if I'm selecting two, you'll probably select the first two. But what we are trying to do is that, can we improve upon this? Okay, so that's what we are trying to do with the simulation optimization. So typical genomic selection process, you'll have the breeding population genotype, and then you put that into the prediction model. And then the prediction model, you have the training population, you'll go through the process, and you'll have a reasonable, accurate prediction model. Okay, I also have some work in, for example, training, population selection. There's some optimization techniques over there, but again, that's not included in today's talk. But today I want to focus on the green highlighted area, which is selection and mating. Okay, so traditional or typical genomic selection, after you make your prediction, what you are doing is that you do truncation selection. Supposedly you can pick the top 10% to advance. You just pick the top 10% based on the prediction model, the output, the predicted phenotype, and then you throw in the rest of them. Okay, one of the biggest problem is the some of the less favorable ones may carry some really important genotypical information, which will be important for further improvement down the road. Okay, so this is what's called the trade-off between short-term and long-term gain. Now I'm giving one example of genomic selection for example, 3 generations. So if you have the first generation, you are selecting, for example, 4 individuals. You selected these 4. As you can see, they are circled. And then the first 2, you cross them, and then you produce 6 individuals, 6 progenies. And then you cross this 2, you produce 5, for example. This is just one illustration to show what kind of decision could be involved and how complicated. Now, in the next generation, you could have back crossing right over here, or you could cross individuals within that generation, or you can do selfing. And then that can advance to the next generation. And this process will go on and on. So when I look at this, in the system design thinking or view, there are a lot of decisions that can be made. For example, in the first generation, which ones to pick, how many to pick, and how do we pair them? How do we pair them to mate? And then when we mate, how many progenies should we genotype and phenotype take to the next generation? And in the next generation, should we bring in the previous generation for back crossing? Or should we actually do some selfing? Okay, so there are a lot of decisions. Think about when you have a large population, the decision space is going to go exploding. Okay. Now, I want to quickly talk about 3 studies we did, and they're sort of connected, built on top of each other. The first one is called look ahead selection. And then the second one is the multi-trait version of look ahead. And then the third one is what we are currently working on, that's called reinforcement learning. So in the look ahead, or in the genomic selection in general, the problem definition is that given the initial breeding population, how many individuals should we select? And how do we pair them up? And how many progenies should we produce in each of the cross? And some of the challenges include when we are really dealing with real breeding, there are a large number of alleles throughout the entire genome. Well, for us, we were dealing with just the data we were provided 1.3 million SNPs. And they may be varying contribution of the alleles, okay? And then recombination, here should be probabilities, recombination frequencies, and then also budget and deadline constraints. You only have limited amount of resources, and then you have a target, let's say, you only have 5 generations or 10 generations that you are making decisions for. Now, for look ahead selection, the concept or the idea behind that is that we're trying to maximize the expected GBV of the progeny in the terminal generation. So the concept behind look ahead is that we're not just looking at what is predicted in the next generation. We know that our target is 10 generations down the road. So what we are doing is simulating all the way to the 10th generation. But if you think about it, that's a very, very complicated process. Okay, so we designed some algorithm to really can look ahead and then use that information to inform our decision making in the current generation. The input would include the typical genotype, recombination frequency, and also time constraints. The output will be the selection strategy and also mating strategy. So typical genomic selection, we tend to just assume they are randomly mating. We don't care which one is mating with which, but actually that could make a difference. And then we do consider how many generations we are working with, and also the mating strategy. And this paper has been published in G3 in 2019. Given the time, I'm not going to go into the detail in the optimization model, but if you're interested in this, you can look into the paper. We provide all the details. But the key point here is that, well, in IE, this is called a optimization model. You have objective function, and which is subject to a variety of constraints. Okay, so I'm using a matrix form, but actually there are many of these type of constraints. I want to draw your attention to the top over here. The decision here will include which one to select, but they are information about how many generations are still left when I'm making the decision, and what kind of recombination frequency we are facing in this type of population. So that's that. And in terms of the look-ahead selection, again, the key idea is that we are at generation lowercase t. And typically what we do in conventional genomic selection is that you genotype, use the prediction model, or you can either simply just use the allele effector to calculate the GBV and make a triangulation selection over there. So what we are doing here is that we are actually simulating the reproduction process all the way to the terminal generation. And then we'll rank them, pick the top few percentile, and then we'll do the evaluation, and then we'll trace back. Again, we are still at this time decision-making point, but we're using some future information to make the decision for the current generation. Okay, so by doing that, as we'll see over here, by doing that, it can actually balance the trade-off between the current gain and also future gain. So this curve shows the cumulative distribution function of the 4 algorithms, okay? So the red one is the conventional genomic selection, and then the black one is the OHV, optimal haploid value. And then the pink one is another paper that was published earlier in our group. It's called OPV, optimal population value. And then the LAS is the look-ahead selection. I want to explain this chart real quick. The x-axis is the genetic gain, and then y-axis is the percentile. So you can think of, if you have a 50 percentile, if you draw a line across, this is about the average genomic of the GBV value in the terminal generation. Okay, so the general rule is that the far right, the curve is, the better the selection method is. Okay, so as you can tell in this simulation, well, this is actually an average of a simulation of 1,000 rounds. So we already consider some uncertainty in the in the recombination process, and as we can tell, the look-ahead selection is performing better. Just real quick, I think I already talked about this. And then for the multi-trait look-ahead selection, what we are doing is that now instead of just looking at one trait, we may be focusing on 2. Okay, so now there are different scenarios you can look at. For example, we are trying to maintain the plant height in a certain range, but we're trying to maximize yield. Okay, so and we extend this to multi-trait, and we benchmark against index selection. And this is all the simulation platform. We designed some algorithm that can look at a variety of scenarios. For example, if you are focusing on certain trait and then trying to maintain some other trait within a range, or you're trying to make sure it is below certain range or above certain range. Okay, so this paper has also been published in Genetics. So if you're interested in knowing more about this, you can either contact me or look at or read this paper. But just real quick, I want to show you some simulation results. I want to draw your attention to this graph. There are two columns here. A column is talk about one simulation replicate. So each color represent one generation. So there is a population for the first generation. So as it goes, so the x-axis is one of the traits, that's the yield. So we want it to be as high as possible. So the far right it goes, the better it is. And then the y-axis is the second trait. So in this specific scenario, we're trying to control it to be within 20 and 30. So that's the first scenario. So we want to breed this population or this type of population to be as high yielding as possible, as well as the other trait within a range. Now, the second scenario is that the initial population is actually higher than the second trait. So we want to bring down certain traits. Let's say we want to minimize something. And then the x-axis, you're trying to maximize yield. And then the third scenario is that we want to bring up the second trait to certain level. Okay, so these are three scenarios that we looked at. Again, each color represents one population for that specific generation. So as you can tell, as the generation goes, the X yield, the first trait yield is improving over generations. And then we are gradually moving to the range that we are targeting in. And then this is one simulation replicate. This is 100 simulation replicate. So when we did 100, we did a mean. We are plotting the mean as well as the standard deviation, okay, the range for both traits. This is for the yield trait and this is for the high trait. Okay, so this slide shows, it's a very busy slide, but this slide shows the comparison between the multi-trait look-ahead selection comparing with index selection. I want to draw your attention to sort of 4 regions. Let's take a look at the first region. This is the comparison between single trait look-ahead and index selection when we are just focusing one. Okay, so this 1, 0 means what's the weight of those two traits. Okay, so this comparison shows single trait look ahead is better than index. And then here I have 3 other regions, 1, 2, and 3. And these 3 cases are corresponding to the 3 cases that I was explaining over here. Okay, so I'll just use one to explain the comparison. So let's take a look at just case 1. If we have this multi-trait look-ahead selection, okay, again, the far right, it is the better. And again, we want to make sure it is within this sort of light greenish band. So as you can tell, the index selection, the best one we analyzed was 0.7 and 0.3 as the weights. And as you can tell, it cannot outperform. So the multi-trait is outperforming the index selection, and same for the other. And some of the cases, it cannot even satisfy the criteria, which is trying to bring up the trait to this light green region. Okay, so again, this is to summarize. And then the very last one I want to mention real quick is a working in progress. I don't have any result to show, but this can show you some of the things that we are working on currently. And in this case, we are adding some additional complications. Okay, so the additional complication is resource allocation. In the previous look ahead as well as multi-trait, we assume that the resource is either distributed evenly or decided ahead of time. Now the question is that, given the budget and deadline constraints, how do we best allocate? What is the optimal allocation mechanism throughout the generations? And so look ahead selection itself is not able to optimize it because the algorithm is so complicated that it cannot incorporate another decision into that dimension. So we are integrating look ahead to this reinforcement learning framework for genomic selection. And I don't think I need to go into too much detail over here, but this is basically saying 2 examples of resource allocation, okay? Like how many progenies you pick and how you cross, how you mate, those type of things. Okay, so just real quick, reinforcement learning. It is a mechanism that's basically talking about how things can dynamic change as things goes by, okay? So some of these are really detailed mathematic formulations. So reinforcement learning technique is formulated in the framework of Markovian decision process, which is sequential decision making under uncertainty. And why is there uncertainty? Because of the reproductive process of the plant, right? And we formulated in this stage, we are running experiment, but I don't have results to show you. So the training algorithm, just real quick over here, this is sort of the framework of what we are doing here. So here we have a deep neural network to simulate the process, okay? And then the look ahead selection, when there is an action taken, is going to be plugging into the simulation. And then the simulation result is come back and inform the forming of the improvement of the neural network construction. So there is this continuous learning process. So to summarize, in the breeding, I've talked about look ahead, multi-trait, and also reinforcement learning. The first 2 have been published. The last one is still working in progress. We have not published it yet. Just to summarize, because when I was first invited to give a talk, I got the advice or requested to talk about system thinking and data analytics. So given the information I provided, I would just want to use one slide to sort of reflect how, or maybe how we can use this type of techniques or concept when we are doing plant breeding. One thing I want to say is that it's important to zoom in the problem and sometimes zoom out to look at what am I doing? How would that plug into the overall process? And that would apply to problem definition as well as solution process. And another thing I want to mention is that plant breeding, my understanding from the system engineering process is that it's a multi-scale analysis and decision making. You can zoom in and take a look at the genetic information, but you can also zoom out and look at how plant breeding could plug into other systems. That's also tied to the very last bullet, which is interactions with other components in the agricultural production system. Okay, so with that, I will stop. I really like this quote. It was shared by one of my colleagues. So what you see depends on how you see the world. To most people, this is just dirt. So 10 years ago, you asked me, this is dirt to me. But to a farmer, this is potential. But as a researcher in this area, it's very important for us to provide our contribution to maximize the potential of this piece of land. So with that, I will stop and answer any questions you may have.